Learning objectives. Understanding the basics of clone culture. Grasp the fundamental concepts of clone culture, including the definition and process of isolating single cells. Identify the key steps in establishing a clone culture from a single progenitor cell. Techniques for single-cell isolation. Learn various techniques for isolating single cells, such as limiting dilution, micromanipulation, and flow cytometry. Understand the advantages and limitations of each isolation technique. Clonal expansion and maintenance. Understand the conditions required for clonal expansion, including culture media, environmental conditions, and growth factors. Learn how to maintain the integrity and purity of clone cultures over multiple passages. Applications of clone culture. Identify and describe the various applications of clone culture in research and industry, such as genetic studies, biotechnology, drug testing, and cell line development. Understand the role of clone culture in producing recombinant proteins and monoclonal antibodies. Quality control in clone culture. Learn the importance of quality control in maintaining clone cultures, including contamination prevention and genetic stability. Understand methods for monitoring and ensuring the purity and integrity of clone cultures. Learning outcomes. Fundamental knowledge. Define clone culture and explain its significance in animal cell culture. Describe the process of creating a clone culture from a single cell. Application of clone culture. Identify and explain the different applications of clone culture in genetic research, biotechnology, and pharmaceutical development. Discuss the production process of recombinant proteins and monoclonal antibodies using clone cultures. Quality assurance. Implement quality control measures to ensure clone culture's purity and genetic stability. Recognize and address common contamination and genetic drift issues in clone cultures. Critical thinking. Evaluate the advantages and limitations of using clone cultures in various scientific and industrial applications. Assess the impact of clone culture techniques on the reproducibility and reliability of experimental results. In animal cell culture, a clone culture refers to a population of cells descending from a single progenitor cell. This process involves isolating a single cell and allowing it to proliferate under controlled conditions to form a large population of genetically identical cells. Key aspects of clone culture in animal cell culture. Single-cell isolation. The initial step involves isolating a single cell from a heterogeneous population. This can be done using techniques like limiting dilution, micromanipulation, or flow cytometry. Clonal expansion. The isolated single cell is cultured in a suitable growth medium under conditions that promote its proliferation. Over time, this single cell divides and forms a colony or a population of genetically identical cells, known as a clone. Homogeneity. Clone cultures are homogeneous because all cells in the culture are derived from the same progenitor cell. This uniformity is advantageous for experiments requiring consistent and reproducible results. Applications. Clone cultures are widely used in various applications, including genetic studies, to study the effects of specific genes or mutations in a controlled and uniform cell population. Biotechnology, to produce recombinant proteins, monoclonal antibodies, and other biologically active molecules. Drug testing, to evaluate the efficacy and toxicity of drugs in a consistent cell population. Cell line development, to create stable cell lines with desired characteristics for research or industrial purposes. Quality control, maintaining the integrity and purity of clone cultures is crucial. Contamination or genetic drift can compromise the homogeneity and reliability of the culture. Overall, clone culture is a fundamental technique in animal cell culture that enables researchers to work with genetically identical cells, facilitating precise and controlled biological studies. Conclusion: clone culture is an indispensable tool in modern cell biology and biotechnology. Its ability to provide genetically uniform cell populations underpins many scientific and industrial advancements. By ensuring consistency, reliability, and precision, clone culture enhances our capacity to explore cellular processes, develop new therapies, and produce high-quality biological products.